Hello, everyone. Thank you for joining us for our meeting. Today, our guest speaker is Tatsuya Sakaguchi. His early career as a scientist, it started as a chemist with the ultimate question, how accumulation of thousands and millions of chemical reactions can become life. He has a PhD in biochemistry from Hokkaido University, and his projects were started with specific protein of interest, but it became into formation of networks of molecular interactions, especially in protein-protein interactions. Right now, he has a permanent position in Kurume University as an assistant professor, and right now he's guest scientist at UDM. So floor is yours. You can start, Tatsuya. Thank you for joining us today.
Okay, thank you, Nazari, for inviting and give me a chance for speaking, talking a project for bioinformaticians. So, okay, can I start my talk?
Yeah, you can share your screen right now.
Okay.
Thank you. Yeah, we see it. It's perfectly fine. Yeah.
Okay. Can you see my slide?
Everything is fine. Yes, we can see it.
Okay. Today, I'd like to talk about our latest project, mapping protein interaction quantitative toward loci. We called this QTL as a piQTL. And this piQTL unveils a novel aspect of gene and environment interactions. Then this project is already published, available in bioRxiv. And then the bioRxiv paper name is this one, Genetic Landscape of in vivo protein interaction. And also we are under revision process for this project for publishing some paper, some journal. Yeah. Then I hope in the near future this project, this paper is available on quite big journal, I hope. And now I'm Tatsuya Sakaguchi, and I'm located in here. And this project is mainly provided by Savandra, Savi, and I. Me and Savi are equally contributed to this paper, and the main supervisor is Adrian Serhijos. So then Savi is correspond to the bioinformatics analysis of this project, and my responsibility is experimental part. But I know today's main guest is based on bioinformatician, so I try to focus on the informatics analysis. But sorry, my mission is experimental part. So if you have some question or lacking information about some part, so please let me know and give me some question or feedback. And also today I have only 30 minutes and I cannot dig in details of this project. So, but I want to guide an overview or a concept of this project. That's what I want to speak. I want to speak today. So then first of all, I want to show the project goal of this project. So the main purpose of this project is providing the concept of the piQTL. So what is piQTL is I will explain by using the following slide. But the piQTL is what kind of QTL and based on protein-protein interaction. Then that's located on the next layer of protein abundance QTL, and that is quite close to the phenotype. So that's our claim, and we try to constructing the experimental pipeline of piQTL analysis. So then maybe you already know, but I would like to start from briefly introducing the what is QTL, quantitative trait loci. So this is quite a schematic image provided from some Nature review paper, and maybe this decade scientists are mainly focusing on the genome sequencing. Details, genome sequence are detailed, and many species or many organisms sequence are available. Then based on this, based on such sequencing information, scientists identifying the relationship between genotype, gene sequence, and phenotype. So in this schematic image, there's two types of leaves. One leaf have a many hair, and some leaf is not so many. Then they try to detecting which site or which sequence of genome sequence are determining the leaf sequence, leaf hairs. So this is a, I think this one is called GWAS, Genome-Wide Associated Study, but essentially this one is one of is deciding the leaf hair amount. Then based on this analysis, we can get many information, and this scheme is focusing on the leaf hair, but this one is easily applied to the disease or sick or many other kind of phenotypes. So someone have some mutations that maybe easy to become a cancer, or someone is opposite. So some mutation is quite resistant to the cancer or some disease or sick. So such a genotype and phenotype relations are revealed or identified by using a next generation sequencing or similar techniques. Then this information is quite important, but still there's quite a big gap between genotype and phenotype. So but like this approach, we can identify the relationship between genotype and phenotype, but this one cannot explain why such genotype make such phenotype. So there's huge gap. Then to try to fill in this gap, the next scientists are try to analyze the next level relationship between genotype and phenotype between here. So ultimate phenotype is leaf hair or disease type or height or weight or eyesight or many phenotypes are expected. But at the molecular level, the next phenotype or output between genotype and phenotype is messenger RNA level, expression level. Then the QTL based on messenger RNA expression level is called eQTL, expression QTL. Then they try to fill in the gap between genotype and phenotype. So this genotype increasing or decreasing the messenger RNA level, then that could be explained why such a phenotype happened. And also similar approach are performed, and many kind of QTLs are created to try to fill in the gap between genotype and phenotype. So the eQTL or protein level QTL or what is the SQTL? So I think splicing variant, yeah. Actually, so many type of QTL are created. Then I believe the main motivation is filling the gap between genotype and phenotype. So they try to this phenotype that can be cause some final output of phenotype. Then this gap is gradually filled. So from genotype to phenotype. Then now we want to make a new piQTL to filling the gap. And I believe protein-protein interaction is quite on the front of this, how to say, of this QTL development. So there's many QTL, and piQTL is one of them. Why protein-protein interaction is so important? Because protein is a main player in the physiological reaction. So there's many chemical reaction in the life is mainly performed by enzymes. So this is also, of course, protein. And also protein have many other roles, scaffold or transactivation or transport or storaging some integrating or oxygen was storaging something, and immune response and muscles or many kind of, there's so many roles of protein. And more importantly, such protein function are mainly depending on the physical interaction. So not only one protein perform a full function. For performing a protein function, they make a complex, or they make such as signal links, or this one is quite relatively simple, but in this one, I think 2 or 3 protein making a complex and do a work as a molecular motor, or many kind of 10 or 20 protein complex is not so rare. So such a which protein make a interaction of which protein is quite important for understanding full protein function. So that's why we are focusing on the protein-protein interaction, not only on one protein. So like that. So then such a protein interaction network called interactome, interaction-based protein society. So there's so many kind of protein complexes, and also such protein complex also communicated with using some interactions. Then such interaction make a quite huge network system in life, and such network is quite depending on this interaction itself is quite complex and huge, but more interestingly, this interaction network is quite dynamic. So dynamic means such interaction is depending on the environment. So if some poison or nutrient carbon deficient or nitrogen deficient has happened, this interaction network is dramatically changed, and they try to adapt such an environmental change. So then, for understanding the life, how interactome network are changed, understanding how interaction network change is quite important. But so far, the genotype and such interactome, the relationship of genotype and such a interaction network is still unclear. Then to understanding this question, or to understanding this point, we are constructing a new experimental pipeline based on DHFR protein can be split two parts. Then, of course, that splitted DHFR cannot work. But if these two splitted fragments are physically located physically close, the full function DHFR can be reconstructed. Then this reconstructed DHFR work as an enzyme. Then this enzyme make a vitamin. So then in this technique, we introduce some point mutation. Then the native DHFR of human cell, or in our project, we use yeast. Most organisms have their own DHFR, but that DHFR can be stopped by adding a drug. In this case, methotrexate, MTX can stop the native DHFR. Then if this native DHFR are stopped, the organisms cannot survive. But if such reconstructed DHFR existed, they can be survived. So then by using these characteristics, we add these two fragments to our targeted protein. Then if two targeted proteins, then if these two proteins make an interaction, this DHFR are reconstructed. Then they can survive. Then by using this, technique, we make a yeast library. This yeast library have many. This yeast library have many variation of genotypes. So typically, for briefly, this yeast have two parents, and they have a different genotype, of course, but they are inbreeded 6 generations. Then the one characteristic of yeast is they are easily homologous combinations. So it means their genome is quite mixed. So then, if this inbreeding, these two different parents' genotype are mixed, and there are many mixed genome yeast cells are constructed. Then in our project, we isolated 357 different yeast strains. They have a unique genotype derived from two parents. So like that. This is kind of a mosaic. One row is one strain. So there are, it means there are 355 rows. And also these two parents have 12,000 SNPs. So then such SNP is mixed or randomized during this inbreeding. Then each 357 strain have a unique genotype based on derived from two strains. And this 12K SNP is located on many genetic features. Then by using this yeast library, we introducing a DHFR fragment for our targeted strain, targeted protein pairs. So we constructed 61 yeast library. So one yeast library have a DHFR fragment for specific protein pair, protein combination. So one library targeting A and B protein are labeled by DHFR. Then next library, this is they have a same genotype background, but the DHFR labeled protein pair is different. Then totally we have one library, two libraries, three libraries, blah, blah, blah. Then totally we have 61 yeast library having a different gene pair are labeled. Then labeled genes like that, and there are 61 different protein interaction are analyzed by using this library. Then the essence of piQTL analysis is we know, for example, this ZTR2 and PMC1 make a protein interaction, but there are 355 different genotype yeast, and we expected such genotype difference make a difference of protein interaction intensity or strength or stability. Then by using this library, we add a metotrexate to stopping a native DHFR. Then if these yeast want to grow, they need to use this reconstructed DHFR. Then if one genotype make a more stable protein interaction for protein X and Y, they can be growth rapidly compared to other genotype. Or opposite is true. If some genotype unstabilized protein X and Y, their growth speed is low. So then such growth diversity could be expected. Some genotype make stable protein interaction, their growth speed is quite high. Then if some genotype make an unstable protein interaction, their growth speed is low. Then during competitive growth condition, these strains frequency are changed based on protein-protein interaction intensity. Then, this is an overview of our project. So we introduced DHFR fragment to specific gene protein pair, and this pooled strain grows in one tube with competitive condition, and we constructed 61 PPIs and plus no PPI control. Then totally we constructed 62 libraries. Then also we growed these 62 library under 5 different drug conditions, also to checking the protein interaction intensity with other methotrexate. Then this is a typical result from our piQTL analysis. So after constructing this library, these strains grows under competitive condition. Then some strains frequency are increased because they make a stable protein-protein interaction, and someone's frequency go to decrease because their protein-protein interaction is relatively weak and stable. Then based on this frequency, each strain's frequency, we calculated relative fitness of each strain, just simply divided their frequency, final frequency by initial frequency, then take a log. Then for compared to the other conditions, we subtract a reference strains relative fitness. So this one is baseline. Then this simple equation make a PPI intensity or PPI based fitness. Then this PPI fitness is classified based on each SNP site genotype. So essentially there are 2 genotypes come from two parent strain. Then each strain's fitness are plotted based on their genotypes. Then this is one of the typical results. For example, this one is result from ARG11 PIS1. This is protein-protein interaction with methotrexate under fluconazole condition. Then this SNP4292 have a quite a big piQTL effect because the strain have a -1 genotype, generally show a weak protein-protein interaction, but the strain having a +1 genotype, their protein-protein interaction is relatively high. So then we can quantitatively analyze their protein-protein interaction from this, based on this analysis. Then essentially we can make this type of plot for all SNPs